Muscular tissue. Property of contractility is possessed by all the living protoplasm. It is particularly well developed in muscular tissue. In such tissues, the direction of contraction is along a definite line which corresponds to their long axis of muscle cells. In most metazoans, all movement is brought about by the contraction of muscle cells. Such contractions are responsible not only for locomotion but also for heartbeat, for the propulsion of blood, and for the propulsion of limb, though the vessels for the passage of food through the digestive tract and for the passage of glandular secretion and excretory product through ducts leading from one part of the body to the another. Smooth muscles tissue. It is made up of a long, narrow, spindle-shaped cells, each of which bears a centrally placed nucleus. A thread-like myofibrils run in a longitudinal direction through the cytoplasm or sarcoplasm, as it is called in case of muscle cells. The myofibrils are thought to be the actual contractile elements. Smooth muscle cells may occur individually or in the form of bundles or sheets. Individual fibers in close association with collagens and elastic fibers are found in such region as the skin about the nipples, the wall of a scrotum, and the villi of small intestine. Bundles of smooth muscle fibers surrounded by connective tissue are represented by erectile pile muscles which are attached to the bases of hairs and cause hairs to stand erect at certain time. In the walls of hollow viscera, they are usually arranged in two main layers called circular and longitudinal depending on directions and their functions. Smooth muscles undergo slow and rhythmic contractions controlled by the autonomic nervous system. Contractions of circular layer cause a narrowing of a lumen and a lengthening of an organ. Contraction of the longitudinal layer cause increase in the diameter of lumen and cause shortening of organ. In this slide of a smooth muscles, they have shown a longitudinal section of intestine, digestive tube. Here you can see a presence of circular fibers and longitudinal fibers. They are all smooth muscles, but these fibers are arranged in longitudinal fashions and these are arranged in a circular fashion. In this diagrammatic presentation you can see the longitudinal arrangement of these fibers and a circular arrangement of these fibers. Because of this both set of muscle fiber, longitudinal and circular, a peristalsis movement is possible. Both in synchronized fashion they contract and relax and we get a peristaltic movement, a passage of food further. Here electromicrograph is shown of smooth muscle cross section and longitudinal smooth muscle cross section. Here a single muscle fiber is cut into a section showing myofibrils and sarcoplasm. Here in a transfer longitudinal sections electromicrograph you can see a nucleus located and the myofibrils and the bands, smooth muscle bands. Here for better understanding a diagrammatic presentation is given of a single muscle cell. A single spindle-shaped muscle cell contains centrally located a nucleus and the remaining cytoplasm is now called as a sarcoplasm with internal supported this fusiform densities alpha actin fibers and mesosine actin myosin fibers. These are actually responsible for contraction of a single muscle cell. Here after contraction longitudinal shortening occurs when these mesosine actin myosin and fusiform density alpha actinin fibers get contracted, a longitudinal contraction shortening occurs and this goes involuntary. it is not under our control. Now, where these smooth muscles are located? In a normal cross section under 48x zoom under compound microscope, you will see a slide of smooth muscle like this, where this pink color portion indicate the fibers and sarcoplasm of muscle fibers, and violet color elongated bead shaped structures indicate nucleus of that muscle fiber. You will find these smooth muscles in a wall of a digestive tract, in a wall of glands, generally a duct. Every gland they have a duct to pass secretion to targeted organ. So in that duct you will find, in the wall of that duct you will find presence of smooth muscles. In all respiratory tract, that is tubes present in a respiratory system, urinary and genital tract, all blood vessels, arteries and vein, then muscles, iris, ciliary body, uterus, bladder, stomach. In a wall of all these organs you will find presence of these smooth muscles. Striated muscle fibers, striated muscle tissues. Skeletal muscles are also called as striated or voluntary muscles. In the development of striated muscle fibers, nuclear division occurs without cytoplasmic division. So the fibers of adult striated muscle is therefore multinucleated. As the nuclei are not separated by individual cell membranes, their grouping forms a syncytium. Each muscle fiber is longer and thicker than the smooth muscle cells and maintain a uniform diameter throughout its length, ending bluntly. Early in the development of the fiber, the nuclei migrate from the center to the periphery of the cell. The sarcolemma is thicker and may easily be seen as a structureless sheet around the fibers. The most striking morphological characteristics of striated muscle is the transfer marking of myofibrils. These fibers are thick and called sarcostyles. Each sarcostyle is composed of succession of bands of different and alternating indices. One set of these is anisotropic and appear to be dark and called A band. Each A band has its middle or somewhat lighter zone termed H zone or Henson's line. Other set is a isotropic and has a pale color. This is called I band. Each I band is crossed through its center by a dark line called Z line or a membrane of Graeus. The dark band alternate regularly with light bands, giving the muscles its characteristic transversely striated appearance. The part of the myofibril between two successive Z line functions as contractile unit called as sarcomere. So a sarcomere consists of a band and half of each adjacent I band. In this slide, a cross section of tissue showing striated muscles are showed. Striated muscles are identified on basis of a very fine and regularly placed dark and light band. This is called lines, striations. And if you trace these striations, somewhere you find this dark violet color spot arranged at the periphery of this line. It's called as a nucleus of, and this is a striated muscle fiber. All these structures, these are also striated muscles, but they are cut in a transverse plane, and these fibers are cut into the longitudinal plane. So here these are all striated muscle fiber cut into transverse plane, and they are showing this nucleus sarcomeres here. Again, a cross sections and longitudinal section of a striated muscle is shown. These are the longitudinal sections where the fibers can easily be seen and the surrounding nucleus are seen here. The same striated muscle fiber when cut in a long sections and transverse sections. A band of a striated muscle is cut into transverse sections. You will find there is a striations clearly visible. So on that basis of striations we can identify these as a striated muscles. Under the compound microscope the slide look like this. All pinkish color indicate the sarcoplasma, cytoplasm, and violet color beads indicate the nucleus of muscle cells. Here skeletal muscle fiber types are shown which are clearly visible in case of a chicken, where in case of a chicken leg you will find there are more number of red color fibers, muscular fibers are there. In case of breast meat you will find there is a more number of white fibers are there. These red fibers they shows slow twitch, less tensions and high endurance. Numerous mitochondria are there, so they look red and they indicate a slow but high endurance movement. In case of white fibers there is a very few number of mitochondria, so they look lighter in color. They shows a fast twitch and they can withstand for to more tensions and fatigues rapidly. So in a cross section you will easily able to notice a presence of white muscle fibers and red muscle fibers. So here these are the white muscle fibers and these are the red muscle fibers. Fibers type stained by this color appear because of a particular staining of a glycogen. Pass reaction is performed over this slide section and it produces this differentiation color. Here a cross section or a photograph of a slide showing the junctions of muscle fiber, striated muscle fiber with the tendon is shown. So here in connective tissue we have seen this tendon slide. So these are the all connective tissue fibers, elastic fibers which forms the tendon. They are joined with the striated muscles and this is a junction where we can differentiate the connective tissue with the striated muscles. Because of presence of these striations we can separate them. Skeletal muscle vascularization. Now how blood supply is provided to the muscles. With using a particular dye, silver dye, you can visualize the all blood capillary structure providing blood to the muscle bundle, striated muscle bundle. So here you can see all these blood vessels innervated to each muscle fiber and providing the nutritions. In electromicrograph, a striated muscle fiber is magnified. You can see here a nucleus which is arranged at the periphery and presence of this A band and I band which we discussed earlier. The detailed structure of striated muscle is shown in this slide. The myofibrils are further separated into smaller subdivision called as a myofilaments. The myofilaments are of two types. The thick filaments confined to the A band contains myosin. They have minute projections and are arranged in hexagonal shape. The thin filaments are present only in I band. They consist of actin, tropomyosin and troponin. The I band have smooth surface and are so arranged that 6 of them surround each A band. This peripheral overlapping of myosin by the actin imparts dark appearance to A band. During contraction of muscle fiber, the position of myosin filament in a A band remain unchanged, but the actin filament of A bands slide further in towards the Henson's line, it is H line, pulling the Z line nearer to each other and shortening the I band and H zone. Such an action in all sarcomeres result in shortening of entire myofibril and thereby of the whole fibers and entire muscles, and we say that muscle is contracted. The close association between muscular tissue and connective tissue is again brought out in a study of striated muscle. particularly in the region where the muscle is attached to bone or cartilage by means of tendon. Earlier we have seen in a slide attachment of muscle, striated muscle with the tendon. A sheet of connective tissue surrounds the entire muscle is known as epimysium. Bundle of muscle fibers or fascicula are surrounded by perimysium, and the individual muscle fiber is enclosed by a thin sheath, the endomysium. In the tendon, the bundles of muscle fibers are intimately interdigitated with the collagen fibers. Elastic fibers may be found in more than ordinary numbers in certain muscles, such as those which cause eye movement and those of a tongue and face. In this slide, you can see ultrastructure of striated muscles, and here a label A band is shown, I band is shown, and how this Z line appears. Striations develop in a cell, muscle cells, which are visible under light microscope. All these striations are these lines. They shows us a striations under the light microscope. Now, sliding filament model or striated muscle contraction, how it happens. These thin filaments are there. These are the thick filaments forming this M band, and this entire a single sarcomere is shown here. This is a Z line, and at these knobs slide over this thin filament, and all the thin filaments are contracted toward the centers, so reducing the Z line and results into contraction. Now, sarcoplasmic reticulum, how nourishment is provided, how these muscle cells are innervated with sarcoplasmic reticulum is shown in this slide. So here these are all fibers of striated muscles sections, and they are surrounded with these different canals, that is T tubes and triad. These all channels over these muscle fibers, they are called as a sarcoplasmic reticulum. Here in this ultra micrograph you can see sarcoplasmic reticulum spread over the muscle fiber. Now all these striated muscles are voluntary muscles, so our willingness, we can contract and relax these striated muscles. So all these muscle fibers are innervated with the nerves, and the area where they attach to the muscles, that area is called as a motor end plates. In this photograph of a slide you can see presence of motor end plates. These nerves are attached to the muscle at this location. The nerve impulse comes through these nerves and gets spread over the muscle fiber, and that results into contraction or relaxation. In electromicrograph it look like this. These are the motor end plates of a nerves over the muscle fiber. This is how these motor end plates are attached to the muscle fiber is shown in this micrograph and the diagrammatic presentation, that is called as a myoneural junction. These are the nerve cells attached to this muscle fiber. Now this myelin sheeted axon, the nerves impulse enter in these myoneural junctions. They are covered with the teloglia, arborization nuclei, and this is a cell of nerve cells. They produce synaptic cleft at the junction, and there transfers neurotransmitter to the muscles, and they transfer that information of contraction or relaxation through this synaptic cleft through the neurotransmitters. These are all subneural apertures which get the information through the neurotransmitter. Biochemical changes happen at the surface and the information get transferred. This is a primary synaptic cleft and this is a secondary synaptic cleft where there is a actual transfer of nerve impulse information take place through the neurotransmitters. Cardiac muscle is a third type of muscle. It is present in heart. The functional peculiarity of cardiac muscle is its ability to contract rhythmically and continuously. The contraction is entirely independent of a will. You cannot stop or start contraction of cardiac muscles. Morphologically, cardiac muscles may readily be distinguished from smooth muscles and skeletal muscles, although it shares some of the same characteristics. In structure, however, the cardiac muscles more closely resemble skeletal muscles since it is striated. In these cardiac muscles also, you will see the striations. Alternating dark and light discs are present in a myofibril. Lines you see that is called intercalated disc. The nuclei of the fibers, however, are placed at more or less regular interval. They present they are located deep within the fiber in its axial portions and are in contrast to the arrangement of a nuclei in skeletal muscle. The fibers branch freely and anastomose so as to form a network of muscle fiber. They are not arranged in a same single longitudinal fashion, but they show branches and they goes in any directions. The sarcolemma is also less noticeable than that of skeletal muscle, where the fibers meet at end to end. Thick cross bands, the intercalated discs, are formed rather like Z line which are present in the skeletal muscles. The functions of intercalated disc is not understood, but the cardiac muscles like the skeletal muscle contract quickly but does not get fatigue. Here in this slide you see intercalated disc. You can say they are sort of attachment of between the two cardiac muscles. A clearly and centrally located nucleus is there and they shows a branching structure. So you cannot say a band of cardiac muscles, but they are branch interconnected. Here in this color photograph of a slide, cardiac muscle slide, you see the presence of these lines. These are the intercalated disc or separations of cardiac muscles and very prominently centrally deeply located nucleus here. Here in this staining you see only the presence of nucleus and sarcoplasm layer is stained very faint. Cardiac muscle fibers in a transverse sections look like this. You can confirm the presence of nucleus at centrally located. This is an electromicrograph of cardiac muscles to visualize intercalated disc. The striations are there, but along with the striations you find this intercalated disc which is not present in the striated muscles, and here presence of nucleus which is at the centrally located. There are gap junctions with the desmosomes. Purkinje fibers, these are the contraction initiating and relaxation initiating nerves like structure present in a heart, that is Purkinje fibers. They are specialized conducting tissue. This is also called as a pacemaker which initiate the contractions or they maintain the contraction relaxation in a heart. Along with the cardiac tissues, along with the cardiac muscles, you will find some different types histological type of a structure in the cardiac muscle sections. So here you can see this Purkinje fiber and surrounding with all these are the cardiac muscles. Purkinje fibers cross section is shown where there is a lot of glycogen store, nucleus and adipocytes are also surrounding these Purkinje fibers. They maintain the contraction relaxation of heart, rhythmic contraction relaxation of heart.